Yeah, exactly. Welcome to our first in-person. For how long? It's been 2 years, and that is funny because that was about when Thorsten first emailed me. I was looking this morning, and it was in early January 2020, and it seems like years ago. It seems like a lot longer than just 2 years ago, and it's great that he can be here in person and that we can have our first in-person seminar. Thorsten's from the Norwegian University of Science and Technology on sabbatical in America for maybe 4 months so far. 5 months. And so that was great timing because we can invite people in to give a talk if he's here, so he didn't have to fly all the way from Europe. He's been enjoying the winter of Southern California, so that has been nice. And this is a nice shop for him, although it's not too cold. And anyway, he's an expert in trying to understand turgor, the maintenance of turgor and the sort of how turgor and cell walls interact and cell walls regulate turgor. And that's how we became acquainted, because we had published a paper on ABA biosynthesis being triggered by a loss of cell turgor. And I incidentally had sent an email to me to my University of Tasmania email, which I occasionally check. And unlike the Purdue emails, which die, I think, as soon as you leave, this is a perpetual email. And I scrolled through and it's like, look, there's an actual email from somebody interesting that I'll read. And it was great to reply to, because it led to some collaborations led to some really great work. So that, I suppose, should be all I should waste time introducing. I guess, but thank you so much for coming, and it'll be a great talk. Thanks for the kind introduction. Much appreciated. So I will try to address the audience in person, the online audience, keep track of my slides. We'll see how that works out. So let me get started. I'd like to get started by, first of all, pointing out the people which actually do the work, because I'm a typical PI, spending too much time in the office and in meetings. So everybody in bold fonts has been contributing to this work. Our collaborators, you may notice Scott McAdam obviously down there. And without further ado, I'd like to get started about talking a little bit about science. So when we watch all these beautiful programs on TV about nature, we are constantly being told that this animal is better than us humans in this respect, and that, and this, and so on. But what I'd like to point out is that actually us humans are extremely good at adaptation to challenges and to different environments. And when you look at these different pictures I've selected here, I think they illustrate this really nicely, because we adapt to extreme environments like cold in Norway, the space, or the beach in Santa Monica. I guess you know what I'm talking about. But we also are very good at adapting to new challenges in the form of COVID. And we are successful in this. We overcome the challenges. Quite often, they may not be very pleasant to overcome. I think COVID has been one of those examples, but we manage, we get through with this. Now, the crucial element here with respect to the adaptation is we often use equipment, right? We don't change ourselves. All these examples here highlight that we use a cocktail, a spacesuit, face masks, and so on. Now, when we look at plants, the situation is different. Plants adapt by changing themselves, right? And I've picked here a couple of examples: California deserts, top left corner, for example, top right, rainforest in New Zealand. At the bottom, you see a cemetery in winter in Norway. And I suspect that when people look at this picture or this slide, and most of us can recognize the topic we are actually working on in our day-to-day life in science, more or less indirectly. Be it drought stress, cold stress,
adaptation to different light conditions, different seasons, and so on. But what I see is actually underlying all these processes, which are the cell walls, because they are more or less directly involved in all of these biological processes. And for example, in the middle, I think the tree is quite clearly indicating from which direction the wind is blowing. I think there's no misunderstanding on that one, right? But when you think about the cell walls in this context, they have to meet different functional requirements. And for example, for the first one, during growth and development, the cell walls have to be plastic. They have to kind of expand, so to allow growth, elongation, etc. When we think about plant pathogen interaction, they have to be responsive. They have to be first kind of involved possibly in perception of the pathogen, and then modification of composition and structure in order to limit the spread of the pathogen to ensure that it doesn't kind of overcome the plants and kill the plants. Or when you think about drought stress, the cell walls have to be kind of reinforced, the stomata have to close to limit water loss, to ensure that the plants have a chance at surviving the drought stress. However, what I'm interested in is actually how do these cell walls maintain their functional integrity during the whole time, regardless if it's being during during development, during biotic stress response, during abiotic stress response. And so this will be a bit the topic of my talk. So how do cell walls maintain their integrity? And I've just spent, I don't know, 3 minutes telling you how cell walls are involved in everything, which makes it obviously a bit difficult to study something which is very cell wall specific. So let me tell you a little bit about our model system. I have to confess, I'm a classic Arabidopsis person, literally. I really struggle with other plant species, but I've realized there are a lot of people here which actually work with proper plants. So I really appreciate that. Now, what we've come up with as a system is quite straightforward. So what we do is we cause cell wall damage by using isoxaban. Isoxaban is a drug that inhibits cellulose production during primary cell wall formation. So it's highly specific in a way, but since cellulose is the load-bearing element of cell walls, it's also hitting the key element of plant cell walls. The nice thing about isoxaban is people have been using it for a while and have isolated mutants which give resistance to isoxaban. So what you see here are seedlings which grow on a plate containing isoxaban at the concentration that we use in our experiments. And the green ones are actually the ones which have a resistance mutation in the CES-A gene, one of the CES-A genes. And the tiny brown dots are seedlings or seeds, which essentially try to germinate and they stop and die because they can't make cellulose. And so IXR1 is always our control group to confirm that the effects that we see are actually specific. Now, to illustrate what happens when you inhibit cellulose production, what you see here is a root tip, which is expressing a plasma membrane marker. And at the top, you see the MOC situation, and at the bottom, you see what happens, for example, to the epidermal cells when you weaken the cell wall. And at the same time, you still have regular turgor pressure levels. So you see swelling of the epidermal cells because they are pushing, essentially, the turgor pressure is pushing outwards. We are fairly confident it is the turgor pressure effect because if you add osmoticum, what you can actually see now is that the swelling has reduced. And this is because we think that it is because the turgor pressure levels have been going down, so they don't push out so hard, so the swelling is limited. Now, what we've done over the last years, we've tried to figure out what actually happens when you impair cell wall integrity or when you cause cell wall damage. And we've spent some time on this. And this illustrates all the various responses that you obtain or that you observe when you inhibit cellulose production. And when you look at this, it's essentially a collection of stress responses, metabolic responses, developmental responses, everything is there. This is not surprising, it's actually expected, because as I said, cell walls are involved in everything, more or less directly. Now, what we've also done as a control, we wanted to see how reproducible these effects are if you cause cell damage in a different way. So what we've taken in this case is we used trisolase, which is a mix of cell degrading enzymes, mostly cellulase, pectinase, xylanase, etc., to see if we can reproduce the effects that we see with the isoxazoline treatment. And we can see qualitatively the same thing, the same effects, at least where we tested, but quantitatively it's not as pronounced. I think isoxazoline is simply more efficient at causing the effect, the cell damage. Now the intriguing thing is, when you do co-treatments with osmoticum, you suppress all these responses, literally. You can really nicely, in a concentration-dependent manner, shut all these things down, which suggested to us that we are looking at an osmosensitive process, which is actually detecting or monitoring the integrity of the cell wall, detecting the cell wall damage, and then initiating the downstream responses. The thing here is, this is obviously all phenotypic characterization. It doesn't tell us anything about the mechanism. So what we've done a couple of years ago, we've actually asked the question, can we identify components, molecular players, which are required for cell integrity maintenance? And what we decided to do is we decided to use a couple of the responses as readouts to then assess the effect of these molecular players on the response to cell wall damage. We used 4 different readouts listed here, and we tested 25 different genotypes. And this is a collection of mutants or transgenic lines, which essentially has been implicated by others in cell integrity maintenance or by ourselves. And we generated a lot of phenotypic data. And the idea was then to cluster the data, similar to what you normally do with transcriptomic profiles, but here with phenotypic data to understand how, if these molecular components are required for cell integrity maintenance, and also because we are using standardized assays, we can put them in relation to each other. So identify the ones which are really having a profound effect and the ones which have less profound effects. And this is what it looks like. So it's a classic heat map. Red means responses enhanced relative to wild type. Blue means responses reduced relative to wild type. The wild type is highlighted with a green arrow in the middle. And for example, at the very bottom, you see IXR1, the isoxazoline-resistant mutant. And this has obviously very much reduced responses because it's resistant to the drug. And what you can see now, we have kind of a couple of nice clustering results. So highlighting here are the ones which show reduced responses. So that means that they're normally required to mediate the response to cell wall damage, to cell wall integrity impairment. And this is a mix of things involved in cell wall integrity or cell wall signaling, but also mechanoperception. On the other hand, we also observe mutants which give us enhanced responses, for example, particularly with respect to phytohormone production. And when you look at the genes you find in there, I think they may be recognized by certain people, BIC1, for example, and BAC1, they are implicated in pathogen response. So you actually get an interesting response here or effect here that if you shut down pathogen response genes, you get enhanced phytohormone production. So what we think is that actually cell wall integrity signaling or monitoring can function as a backup system if you lose your standard pathogen defense response systems. And this seems to be modulated via PEP1, PEP2. I'm not showing the data now, but there's a paper in Science Signaling which you can look at. We can very nicely show how actually PEP1, PEP2 based signaling processes seem to coordinate between PTI signaling and cell wall integrity signaling. And I think the other thing I'd like to point out here is a particular kinase called PCOS1. PCOS1-1 allele, at the bottom in blue, is a loss of function allele. So you get reduced responses, but at the very top of the clustering, what you see is PCOS1-4, which is actually a gain of function allele. And that has enhanced responses. So we have a very nice system here to actually test how this particular kinase, clinical kinase, is modulating the response to cell wall damage. because we can either get the reduced response or the activated response. So this data set has led us to kind of come up with a model, or at least a description of what we think is going on. And I'm not going to go into details here because it's a bit complicated. I just want to show you this. So what we are looking at is we normally have a cell which is happy. And then what happens is it can either experience hypoosmotic stress, hyperosmotic stress, and if it gets really bad, plasmolysis, essentially pretty much close to death, or cell wall damage, distortion of the cell wall. And what we are thinking is that the two extreme situations are being detected or monitored by two different systems. On one hand, we have ABA-based processes, which are involved in detection of hyperosmotic stress and plasmolysis. And on the other hand, we have the crinkle kinases, which seem to be involved in detection of the expanding cells. And when we take a step back and think about it, I think there are two things involved in here. So on one hand, cell integrity, monitoring and ABA signaling. And on the other hand, turgor pressure and cell wall stiffness. So essentially how the cell wall is doing. Now what we've done for the last couple of years, together with Scott, is trying to address these questions. Does our kind of hypothesis, the crinkle kinases and ABA signaling actually modulate these biological processes? Now, when you think about analyzing stiffness of biological materials, over the last 10, 15 years, quite a few different technologies have been developed. And they all have their strengths and weaknesses. For example, atomic force microscopy, great for producing results, great for producing results above the surface of a structure. But internally, you can't really measure below the surface with atomic force microscopy what the mechanical characteristics of the tissues are, the cells are. And what we found is there's another technology called Brion microscopy, which has been developed or observed, invented nearly 100 years ago. But for the longest time, it was only used for material sciences because the hardware was not really suitable for biological living organisms. Over the last 10 years, the technology has advanced. So we can now these days actually use Brion microscopy to investigate the stiffness of cell walls, extracellular matrix, etc., in biological materials. And the nicest thing about it is, if you combine a Brion module with a confocal microscope, you can actually correlate stiffness measurements or coordinate stiffness measurements with localization of fluorescently labeled tags. So for example, looking at the same time at the cytoskeleton and the stiffness of the adjacent cell wall, or the signaling molecules, D2S1, etc., and the cell wall. So you really take a step forward. To give you an idea what this can look like, so first of all, this is the setup for the Brion microscope. And the nice thing about this is, as it's pointed out, you can simultaneously get a wide field image, just the normal structures. In parallel, you get your fluorescent labeled signal, or from your fluorescent label tag. And third, you get the data for the stiffness. So you can literally nicely overlay this. I'm going to show you one example how this can look like. So what people have done here is they have taken expansins, which have been implicated in cell wall loosening, and have put them under an inducible promoter. What you see at the top, 8-4, is uninduced, and 8-4 plus dexamethasone is the induced situation. And on the right-hand side, you have the kind of quantitative data. But what it boils down to is that apparently in the expansin activated or treated cell walls, you have increased stiffness, which as a cell wall person initially doesn't make sense, right? Because you increase the amount of protein which is loosening the cell wall, but at the same time, you increase the stiffness. For a person working on cell wall integrity, it makes total sense. Because what actually happens is the cell detects the loosening of the cell wall, initiates a compensatory reaction. meaning it deposits other cell wall components in order to compensate for the loss of the cell wall integrity. And this can be pectins, this can be hemicellulose, this can be other things, or possibly even cellulose. Okay, so what you get with this result is you can detect changes in stiffness, but you don't actually know what the compositional changes or structural changes are. That means you need to do this with other analysis or analytical methods. Now, what have we done? So, I do apologize in advance. I think some of my experimental setups are very simple, straightforward, and perhaps over-detailed, but I have a track record of losing my audience. And I'm trying to address this by really being clear and transparent. So because we had no clue initially what really kind of to expect, we had a very simple setup. We took Arabidopsis seedlings, we grow them in liquid culture to really control the environment tightly. And we treat them for 6 hours because we know after 6 hours we observe some lignin deposition and production of phytohormones. And so we stuck our seedlings under the Rio microscope. And then what we did do is we measured initially along the root, which is highlighted by the blue arrow, because we wanted to figure out in what area we actually may see differences. And we measured every 15 micrometers one measurement. And we did this with multiple seedlings to get essentially reliable, reproducible data. So what I'm showing you now are the results of measurements in 3 different layers. So we measured in the central cylinder, in the endodermis approximately, and in the epidermis. After 0 hours, baseline, before we start the treatment with isoxaben, and then after 6 hours again, when we expect to see differences. And the area where we think we see differences is in the central cylinder. And that seems to be approximately at 450, 500 micrometers from the tip. And this is highlighted on the scheme on the left-hand side with the red arrow. Okay. So we decided that we do all the following measurements, essentially in the area of the red arrow across the root. So what you will see is literally cross sections, or the data from cross sections of measurements across the root. Now, let's get started. What we did do then is the proper experiment. Again, 6-day-old seedlings, but we took wild type. We took the isoxaben-resistant mutant, so we have our proper control. And we also took TCOS1 alleles where we see either loss of their function or overactivation of the function. And we also treated again NOC, isoxaben, osmoticum, isoxaben. And this is called a lot of work. So Laura, the first author on this paper, spent quite a few weeks in Lyon doing measurements. Now, what did we find? So as I said before, we measured across. And the first data I'm showing you is the wild type. So this is just the mock control, the MSO. And what you see is you start on the outside, you measure across, and you find increased stiffness in the area where the xylem is, which is what you, or central cylinders, which is what you would expect somehow. Now, we are not saying anything about the composition of these cell walls, okay, as I said before.
Well, a question.
Yeah.
So these are intact roots, right?
Yes.
So as you're going across the root, how do you, I mean, you have the contribution of the outer tissues the whole time. So how do you separate out the contributions of the outer tissues to that signal that you're highlighting in the middle?
I'm not sure.
The imaging is going through the epidermis.
Yeah, we start at the outside of the epidermis on one side, and then we measure across. So we measure everything, epidermis, endodermis, cortex, pericycle, central cylinder, and then the same tissue again on the other side. Right?
But it's not section material, it's an intact root.
No, it's an intact root. And this is one of the killing arguments why you want to do real microscopy, because you're measuring the in vivo situation. You are not measuring that material.
But when you're measuring the central cylinder, your laser is going through the epidermis and all these other cell types as well. They're still there.
It's a confocal setup, so you don't really, the outside is not integrated in this, right?
So you can control, okay. So the confocal is on the emission side.
Yeah.
Okay. All right, thanks.
Okay, sorry. And now what you can see is in the isoxaban treated one, the level of the stiffness has reduced in the central cylinder, which is what we would expect if, for example, the stiffness is mainly driven by cellulose. Okay. Now, then we got a bit of a surprise. If you do the same with sorbitol treated seedlings, where you have reduced the turgor pressure, what you actually see is that apparently the stiffness in the central cylinder area is also reduced. There are these discussions that actually the cell wall and cell wall stiffness is directly or indirectly controlled by the turgor pressure levels. So what we see here apparently could be supporting this hypothesis, that you actually, if you lower the turgor pressure level, the cell wall stiffness is also kind of being lowered because there's not so much pushing anymore. Now, the interesting thing is, if you combine the treatments, what you actually see is that you get stiffness levels which seem to be quite similar to the mock situation. So indicating or suggesting that it's like a relative process, not an absolute exercise, and would also be in accordance with what we see when we give our osmotic support to the isoxaban-treated seedlings, where we get the suppression of all the effects. So it looks like a bit like the cell wall things or the cell things. It's quite normal. If you do the same experiment with the isoxaban-resistant mutant, what you observe is that actually the stiffness in the mock-treated situation is elevated compared to wild type. And this is, I think, again, part of a compensatory reaction when you reduce the cellulose. So other cellular components are being integrated. You see a bit of a decrease with the isoxaban, but you see the same response to the sorbitol, like with the wild type, suggesting that the IXR mutation is not affecting this response, which you would expect, because it's not a signaling mutant, but a biosynthetic mutant. And you still get a bit of a elevated stiffness again in the double treatment ones. Now, interestingly, when you take the TCOS1 loss of function, you see a qualitatively clearly different response. So with respect to DMSO versus isoxaban, there isn't really a difference anymore. So it seems not to respond. When you look at the sorbitol, it's still responding like the wild type. And when you look at the combined treatment, it's again still responding. Interestingly, or possibly even more. Interestingly, when you have the overactivated version, the isoxaban treatment, but it seems to affect the response when you have the combined treatment. So what we think we are currently looking at is a situation where two different signaling processes are feeding into the same downstream readout, the stiffness that we are measuring. So you have on one hand, cell wall signaling mediated by TCOS. And on the other hand, you have the osmotic effects. Yes, TCOS is not really directly involved, but possibly indirectly influencing this. So to summarize, what have we learned? I think that we can use microscopy for analyzing stiffness changes in cell walls in plants. We do see effects of the mild plasmolysis conditions on the cell wall stiffness, which I think is really nice, really neat. And the isoxaban treatment induces reduced stiffness in the central cylinder of seedling roots. What we also think we've started to see is that the combined isoxaban sorbitol treatments induce enhanced stiffness in the central cylinder, and that TCOS1 seems to be modulating the response or the changes in stiffness upon isoxaban treatment. This has been a bit complicated, the last slide. I think this is a good spot to stop and ask for questions. there are no questions,
Yeah.
Yeah.
Can I ask another question to the end of the seminar?
Sorry, can you repeat the question?
Yeah, so this is one as a target, as a receptor kinase, what are the targets of it? Like it will be sensitive?
So the question is about I'm repeating for the audience in the internet. The question is about TCOS1 and what we know about targets and binding. So TCOS1 is a crinkle kinase sitting in the plasma membrane and has an extracellular domain, possibly binding pectin, because another family member has been shown to, at least in vitro, bind pectin. It has been shown to bind RALF34, a peptide, in a pH-dependent manner. And we don't really know how it is detecting or not detecting cell wall integrity. I think that's the $64,000 question.
Yeah, another question is, are there sensitive trafficking phenotypes in the pixels 1-1 and pixels 1-4?
I think nobody has looked, but I'd be surprised. But that's my personal opinion. Because I think TCOS1 seems only to be active or relevant if there's stress or damage, because only then, as far as we can tell, the expression is really activated. And when you look at the growth phenotypes of TCOS1-1 or 1-4 seedlings, they are completely normal. Like we've looked at root growth, elongation, and everything looks fine. There's nothing there. And we also looked at cell walls, and we didn't find any difference in cellulose levels in seedlings or leaves. So, yeah.
Is the Beryllian method calibrated? I'm not sure what the y-axis is on those plots. Is that?
Oh, that's the Brion contrast. Let me see. Here we go. Yeah, so this is Brion elastic contrast, and this is based on the formula I've shown before. Here.
So there's nothing like a modulus.
No.
It's uncalibrated.
Exactly.
So how do you compare it with other methods?
I think at this point, what you would need to do is we haven't done this. You measure essentially, for example, the epidermal cells or cell walls, and then you measure the same epidermal cell walls with AFM. So what you're doing is you are actually getting different types of modulus. So with AFM, you get your Young's modulus, whereas with your Brion, you get the longitudinal modulus. So this is why I'm literally talking about changes in stiffness. I'm not making any kind of statements beyond that. Okay.
So we have a couple of questions from the online audience.
Okay.
Based on what is known about thesis-1 or other characterized cell wall proteins, are there any of these proteins uniquely or especially expressed in the central cylinder?
Can I delay that question until the next slide?
Okay. And one more question. How do you differentiate stiffness due to Turger adjustments versus innate stiffness of the cell wall? Innate stiffness of the cell wall.
Can you repeat that question?
How do you differentiate stiffness due to Turger adjustment versus innate stiffness of the cell wall?
The thing is, we measure after 6 hours of sorbitol treatment. So we would expect as far as I can tell, what we are measuring with the DMSO, we are measuring the combination where the Turger pressure is present and the stiffness of the cell wall is there. With the sorbitol treatment, we would be measuring just the stiffness of the cell wall. But interestingly, when you look at the isoxaban sorbitol, we actually see a change compared to the sorbitol alone. So there seem to be active processes which modulate and drive the stiffness changes. So this is why I think we are actually detecting with the method active modification of the stiffness and not just an effect of removing the Turger pressure. Okay. Good. Okay, good. Now, obviously, the question is a little bit, we have a readout, stiffness, but we actually don't really know how we get from the initial cell wall damage to the downstream readout. So what we've done is we've looked at different reporters where we know that phytohormones, for example, like jasmonic acid, we've shown this before, are activated or induced after cell wall damage. Then obviously the question is, do we actually have expression of the relevant gene in the area of interest? So we have used the TCS1 promoter reporter construct. And last but not least, sorbitol osmotic stress involves normally ABA production. So we decided to look at the response or the expression of an ABA reporter, graph 18. So let's get started. So what I'm showing you first is the expression of the promoter reporter construct for TCS1 under the 4 different conditions in the root tip. And as we can tell, as far as we can tell, TCS1 seems to be induced by the azoxygen treatment in the area of interest, kind of late elongation zone. It's not really induced by sorbitol treatment. And interestingly, when you look at the double treated route, you see that the expression is significantly reduced compared to azoxygen alone. So it seems to be really kind of responding the way we would expect it to. Now, when you look at just 10, you see a very similar situation. Nothing in mock-treated situation. If you give isoxaben, you get activation. It looks a bit like it's in the central cylinder. Surrounding cells a bit. And if you do treat with sorbitol, not much happening. If you do the double treatment, again, suppression. So in a way, it's following what you see for TCS1. When you look at graph 18, the situation is different. You have a little bit of expression, I think, in the azoxygen-treated situation. You have a lot of expression induced in the sorbitol, as expected. And there seems to be still expression in the double-treated, which is to be expected because the double-treated situation, you still have the sorbitol, which is still causing mild hyperosmotic stress. So you would still expect to see this response. Okay. Now, the nice thing about these promoter reporter constructs is you get a spatial idea. Where stuff happens and when it happens. But it doesn't give you anything kind of about the quantitative changes. So what we've done is a different experiment. We've taken seedlings like before, and we've taken, again, TCS1, TCS 1-1, 1-4, did the same treatments like before, and did a time course, because we want to see how the different phytohormones change in these seedlings over time. And measure this. And this is the jasmonic acid. What you see quite nicely is that you have your wild type control called 0, and you have the 2 alleles for TCS1, loss and gain of function. And DMSO doesn't really have any effect. Sorbitol also doesn't induce jasmonic acid. When you look at isoxaben, you see that there's induction in the wild type. There's less induction in the TCS1-1 loss of function. And there's more induction in the TCS1-4. And interestingly, when you look at the double-treated seedlings, isoxaben, sorbitol, nothing, blank. So what you would expect to see if TCS regulates jasmonic acid. And then we got a bit of a surprise. So same experimental setup, we were measuring ABA. As expected, nothing happening, DMSO and isoxaben. However, when you look at the sorbitol data, you can see that in the wild type, you have nice transient induction. I think this is also, for me, a good sign that our mild hyperosmotic stress is not killing the plants if they kind of go back down. But interestingly, with the TCS1 loss of function allele, you get enhanced production of ABA. For the TCS1-4, it's not as clean or as clear, but it seems to be slightly reduced. And the same situation when you look at double-treated seedlings. You still have enhanced the TCS1-1 and possibly a little bit of TCS1-4. Okay, so suddenly TCOS1 is not just a regulator, a positive regulator of jasmonic acid production, but it seems to be also modulating the production of ABA, but as a negative regulator. Now, we all know phytohormones. I was discussing today, everything is connected with everything, and you don't know how direct or indirect it is. So what we reason then is, okay, let's see what happens if we actually modulate the production of ABA and JA and see if there is kind of crosstalk. So how to test in a way how direct or indirect the effects are that we see with TCOs. Okay. So what you do is you do the same experiment like before, but you add a couple of genotypes. You add things which affect JA production, AOS. It's a loss of JA production. And you add ERA2 and ABA2, which are things which affect ABA signaling and ABA production. Same experimental setup as before. You measure, and this is the JA data. And so what you can see is how the different genotypes actually affect the production of JA. And what we see is DMSO not really anything happening, sorbitol not really anything happening, isoxazoline sorbitol nothing happening either. The thing is, this stuff like the differences that are detected statistically, you're at the level, at the limit of detection. You're talking like 10 nanograms or something like that. So the math still finds similarity or differences, but I'm not sure how biologically relevant this is. Now, when you look at the isoxazoline situation, you see nicely that we still have induction in the Columbia. We don't really have much induction in the loss of function of TCOS1, dramatically enhanced in TCOS1-4. And there are some effects of the ABA mutants on JA, but they are significantly less pronounced than the TCOS1-4. So I think there literally are qualitatively fundamental differences. And obviously, reassuringly, our AOS mutant, JA deficient mutant, is JA deficient. Always good when it works. Now, when we look at the other way around, ABA, so not really much happening with the DMSO and the isoxazoline. With the sorbitol, TCOS1-1, dramatically enhanced. TCOS1-4, similar ballpark like before. You have your ABA deficient mutant, nothing, signaling, and a little bit enhancement in the AOS. So we think there is actually some crosstalk between JA and ABA, which has been described before. But I think the crucial message here is, again, the qualitative difference between the TCOS1-4 and the effects that we see with the JA mutation. Similar situation. Good. Now, at this point, I think it's pretty obvious that there is something going on with cell walls and ABA. And this is the part where Scott came into play. Because I'm not an ABA person, I'm not an expert. So what we did do is, what Scott did do was, he did some really cool experiments. So what he did do was, he took Pisum sativum leaves and Arabidopsis leaves, because we wanted to see if we actually something outside of Arabidopsis has the same effects. And he did 3 different treatments. So MOC, our control. Then he did pectinase treatment, which helps to separate the cells, but leaves the cell walls reasonably intact. And he did cellulase pectinase treatments, which essentially remove the cell walls, so you end up with protoplasts, just essentially membrane. And then MOC treatment, all these, and slight hyposmotic shock, the same, or hyposmotic shock, I would say. And then we measure ABA. And what you see here is, I think, one of the nicest data sets I've seen in a long time, to be honest. So, y-axis is ABA, and x-axis are the different kind of samples. You see in the leaves and the isolated mesophyll cells, both for P and for Arabidopsis, that after hyposmotic shock, you get induction of ABA. But when you look at protoplasts, essentially no cell walls, you don't get induction anymore. So we think that you actually need some form of interaction between the cell wall and the plasma membrane in order to induce the ABA production. So this is just summarizing what I've been saying or trying to convey for the last couple of minutes. So that BCOS1 seems to be a strong negative regulator of ABA production and a strong positive regulator of JA production. That ABA acts as a negative regulator of JA biosynthesis induced by cell damage, while JA seems to be a negative regulator of ABA. Key message: we need this interaction between the cell walls and the plasma membrane in order to induce ABA production. Any questions?
I'm curious about the quality in terms of what type of cell wall. So I noticed that all these changes happen mainly on the elongation, differentiation region, not.
So we get these effects because the drug inhibits the cellulose production in elongating cell walls, which happens primarily in the elongation zone of the root. So in a way, we are selecting for cells which are actively elongating, producing primary cell walls. We haven't done these experiments for secondary cell walls because we are struggling to find a good inhibitor for which specifically affects the secondary cell walls.
Yeah, actually, I was wondering if these effects were happening on cells that have secondary cell walls.
I think when you look at the literature, you will find there's a, how to call it, spectrum of opinions. Some people think it's not happening because the secondary cell walls are already so reinforced that it's not really they are not as plastic as the primary cell walls, so the effects are reduced. Other people do think that's the case because they think that there are elements in the cell walls which are being degraded or can act as sensors like pectin in order to kind of indicate occurrence of cell wall damage. I think the jury is out and hasn't reached a verdict yet. And we haven't had the manpower so far to actually also look at that. But that may change now.
So use functions as sensor for cell wall.
Sorry, I'm laughing because that's the big discussion at the moment. So we have these crinkle kinases, and several of them have been suggested to be sensors for cell wall damage. So for example, in our system, we've looked at two of them, Feronia and BCOS1. And what I find interesting is that when we study the responses with BCOS1, it actually seems to act as something required for detecting of cell wall damage, because the loss of the protein leads to a reduction in, for example, jasmonic acid production. There's the gain of function allele leads to an increased production. When I look at Feronia loss of function actually leads to a pronounced increased production of jasmonic acid, which would indicate to me that the cell is still capable of detecting the cell wall damage. It doesn't need Feronia. So currently, I think that both proteins are involved in modulating or translating the response to cell wall damage, but I'm not convinced that they are cell wall integrity sensors. They have extracellular domains which could bind to components of the cell wall, but we haven't really seen this shown or confirmed in vivo. There's some in vitro data for Feronia, but I don't know. I currently would assume that we are looking at a situation where you have multiple systems which detect, for example, a change in the surface tension of the cell walls, or displacement of the membrane versus the cell walls, or something like that, or a combination of both. For example, in the yeast system, you have a combination of both, like detection of changes in surface tension but also displacement and stretching of the membrane. I think we need to do more experiments, but we now start to have the tools to actually look at this.
So what would happen if you just bent the root? I mean, just cause mechanical stress.
We haven't done the experiments, but I would assume the problem is your readout. What's your readout for this?
Well, expression.
Yeah, you could use different reporter genes. Absolutely. My current impression is that there is a significant overlap between what I would cell cell integrity and kind of perception. And I'm not sure how you or if you can actually separate the two from each other. And this is what I'm trying to tell the community, but I'm not sure if that's true.
And this is a stupid question, but
Go
if
ahead.
you actually just tried,
I'll be happy.
could you tell, are they more bendy actually when you treat?
I think they actually, when you look at the roots, they seem to make a turn actually, if you treat with abscisic, but we haven't quantified that. But it seems to be quite reproducible because when we try to take pictures of root tips, it's very difficult to get straight root tips in the treated seedlings. So I think that there is something going on, but we haven't quantified it. It was not the most urgent. Sorry. Yeah.
Is Theseus1 interacting with the stretch activated channel?
That is a brutally good question. We don't know, but we are currently doing proteomics to see if we find something relevant. Yeah.
Two questions regarding the JA elevation after abscisic treatment. So do you see JA elevation after treatment of other cell synthesis inhibitors?
Can you repeat the question?
So do you see JA elevation, increased JA production after the treatment of other cell synthesis inhibitors?
We haven't done the experiment, but so the question was if you see JA elevation after treatment with other cellulose biosynthesis inhibitors. We haven't done that experiment, but what people have done is they've looked at JA levels in mutants which are deficient in cellulose production, which also affects like primary cell wall formation. And there's also an increase in JA production, JA levels.
Follow-up question would be like, what is the mechanism of cell wall damage leading to JA production?
So what is the mechanism of creation of cell wall damage after isoxaben?
What is the mechanism of JA production?
Yeah. So what we think at the moment is the situation is you have a normal cell wall, you give isoxaben, and that means that you lose some of the cellulose.
Yeah.
At the same time, you still have the strong turgor pressure. So that means that suddenly the turgor pressure, which was before contained, is now pushing against a weakened cell wall, and that leads to expansion. We currently don't know if essentially the same change in surface tension or the membrane stretch or a possible displacement of the membrane versus the cell wall could be the stimulus which activates the JA production. This is why, for example, the question about the stretch activated channel was perfectly relevant, because if the membrane is being stretched, you could imagine one of these channels being relevant. So what we found is that MCA1, which is plasma membrane loculated and stretch activated, does have a role there. So this is why we think that we haven't resolved that question yet. Yeah.
If you add the chemical to cause the breakdown of the cell walls and then remove it, do those cells return to their previous, are they reined back in, or would cell walls form in like a deformed way around them?
So the question is, have we done the experiment where we treated with the drug, the cell walls were expanding, and then we removed the drug and watched if the cells are returning to normal? I think that at this point, the cell walls which have been formed are weakened. And we haven't really done the washout experiments to see if they can recover. I think they should be able to recover if you do it in tissue culture, because normally the limiting factor is that they normally, under normal circumstances, die before they can recover. But if you are nice to them, they should be able to recover. Any other question? Okay, going on. So taking a slight detour, because obviously T-series1 is important, but it's not the only thing. So what we've done is we've done transcriptomics. And the nice thing with our experimental setup, as I tried to convey before, is we can cause servo damage and activate responses, but we can also do the opposite effect, shrinking of the membrane by slight hyperosmotic stress. And by combining both treatments, we can suppress the responses, which means you have a very nice setup to do transcriptomics. Try to find genes which are responding to this. And what we've done is we've essentially used criteria where we said, okay, if you treat with isoxaban or with sorbitol, you get changes in transcript levels. But if you do the double treatment, you don't really get changes in transcript levels anymore. And that led to a rather short list of candidates. And for example, for ProPEP1 and ProPEP2, we showed in a different paper already that they are involved in coordinating PTI and cell integrity. So that response or that approach seems to be working. Yeah, okay. So in this case, what we did do is we looked at RP12, receptor-like protein 12. And it's what I would call a fairly large protein and a member of a fairly large protein family. And the first thing we did do was test if actually the changes in expression are specific that we observed with transcriptomics. So time course experiments, both in wild type and the isoxaban resistant mutant, very nicely inducible by isoxaban, no doubt about that. Obviously, this doesn't give you spatial resolution. If you do a promoter reporter construct, it seems to be coming up in the areas where you where we've previously seen the expression of PCOS. We then obviously got knockouts. I'm showing you data for one allele. We've got 3 reproducibly similar results. And it seems that the JA induction is reduced in the RP12 background. And the ABA is unfortunately not affected. So we think that we are looking at that RP12 is only required for modulation induction of JA, not for the ABA effects that we observed. But I think the crucial message here is, again, we can, with this approach, identify novel players that are involved in cell integrity maintenance. I'm not showing you all the other data we have because it's interesting but not exciting, and it's getting a bit late. So the model. Obviously, we have to close with the model. So when we look at this, when you think about cell integrity, as far as we can tell, it's influenced by compositional structure, but also by cell damage. And we think that T-series1 has a key function there. And this function means also that it is modulating the servo mechanics. And this is the stiffness or the data, the results of the stiffness measurements that we've done. What we also think is that turgor pressure influences servo mechanics. Again, the results from the stiffness measurements. We do know that the servo-plasma membrane interactions are really important for the induction of the ABA, which brings ABA into play. And at the same time, we've shown that ABA is repressed by TCRS action, and that TCRS is required for JA induction. And last but not least, we've introduced RP12 as a new player in servo integrity maintenance. Now, why is this so important? When you think back to the beginning of the talk, I've showed you this rather complicated overview. And when you now think about the model I just showed, it's in a way the core element underlying all these processes. Because what you want to do is you want to have a mechanism which enables the cell to monitor expansion or shrinking and have a control element that is capable of integrating the 2 different inputs. And this is what we have at TCOS, because the TCOS is actually modulating the induction of jasmonic acid upon expansion of cells, whereas it's also inhibiting ABA production and can modulate the kind of opposite situation. Okay. And I think this was my last slide. Yes. Are there any questions? I guess we've covered most of the questions before.
One additional question. So maybe have to ask after we see.
No, that's okay. Bring it on.
So earlier in the presentation, you showed a heat map of 4 different phenotypes of different mutants. So like, what is the reason to select this for phenotype like KA and or stiffness?
So what we wanted to do with the 4 different phenotypes is we looked at. So the question is, why did we select those particular 4 phenotypes to do the phenotypic clustering with the 25 genotypes? So we picked JA and SA because they're very good readouts for stress responses. And we picked lignin as a simple way to assess the impact of cell wall composition and structure. And we looked at root growth inhibition as an, on one hand, looking at it from a developmental perspective, but on the other hand, to simply test if there is resistance caused by any of these genotypes that we use, which would influence everything else, obviously. We don't want to just do another IXR1 in a way. Sure.
So Nick Carpita does have his hand raised. Dr. Carpita, you can try to unmute yourself and ask, and we'll see if we can hear you.
Okay, can you hear me? Can you hear me?
Oh. Can you enter your question into the chat?
Yeah, I'll have to do that. Thank you. No, that's really hard. It'll take me a few seconds. You're going to go ahead and have him answer other questions.
Give him a second to type in his question.
Yes. Yeah, I think that was not the intention.
Okay. Oh, there you go. So yes, this new question came in
Yes.
from Noel.
How does this model of T-cell That is a really good question about drought versus pathogen resistance. I'd like to do the experiment. Sorry. Yeah. I think one of the discussion points that we have more recently is when you look at the work that has been done in the cell wall field for the last 15, 20 years, we spend a lot of time trying to modulate cell wall biosynthesis, to facilitate bioenergy production, to improve food crop resistance, etc. And there were some really cool results, really big breakthroughs, but there was also a lot of stuff where we really were quite confident we were manipulating the metabolism and still didn't have any effect. So I'm currently wondering and starting to become more kind of accepted if a large number of these attempts failed because the cell wall integrity maintenance mechanism was monitoring the state of the cell wall, detecting the consequences of the metabolic manipulation, and then going in and neutralizing them. So I think what we actually would need to do is we would need to create lines where we have knocked out the cell wall integrity monitoring and then manipulate and then see what we get. So that possibly quite a few of the phenotypes, mutant phenotypes where we thought we get something interesting, we actually didn't get something interesting because the plasticity of the cell wall or the cell was intervening and messing us up. So, so that is stuff which hopefully we are doing now. So we'll see what happens.
And Nick's question's up on the screen.
So yes, how do your data relate to the PCOS1 based on focused background where cellulose is inhibited in etiolated seedlings? Sorry, etiolated hypocotyles, but growth is restored. I think that when you look at the restoration of the growth, it's actually limited. And I think the thing is that obviously, if you shut down cellular integrity signaling, one of the effects of cellular integrity signaling could be stop in elongation because the cells sense we are not really intact, we are not okay, we need to stop. In the cellulose or in the profuse background, the seedlings have been growing and adapted to the different situation. Like, let me rephrase this. When we measure cell wall composition and structure in ABA mutants, which are deficient in ABA, we actually find a reduced cellulose level. So it seems that essentially in the ABA, the cell wall has adapted to a change in ABA level. What I currently assume is that with the Procust, we have a similar situation, just the other way around. That essentially the seedling has adapted to the reduced cellulose production, and that therefore, essentially the PCOS has only a limited effect. But this is guessing. Any other questions?
I have a related question.
Yes.
Can you remind me, so it was, PCOS was found as a suppressor
of Procust.
Okay.
Yeah.
So does that make sense?
Yeah, the thing is, what you want, what apparently is the conclusion is that you need active shutdown of cell elongation growth. And this is where PCOS comes in. Okay. So
it can't actively shut it down.
Anything else? Okay, good. Thanks for coming for the in-person seminar.
Change your